and start recording. So we were talking about proteins and amino acids. And so amino acids are the building blocks of proteins. And we're kind of deep diving rapidly into biology and biochemistry, right? And so the amino acids are going to represent your sort of vocabulary of proteins. They would be like the words that compose your sentences, right? That then compose your legally binding contract or your publication that you're reading. Okay. And so the amino acids are going to have 3-letter and 1-letter abbreviations because these things can be extremely long, right? You can have proteins that are going to be thousands of amino acids long. And so if you imagine writing out the full name, that means over and over and over thousands of times. That'd be crazy, right? There's no way you're going to do that. It's just way too long. And so, of the 20, 11 are going to be 1-letter abbreviations that start from the first letter of the name of the amino acid. After those 11, we ran out of those first letters because they had a lot of them share a first letter, right? And so what ended up happening is the 9 amino acids are going to have 1-letter abbreviations that start with a different letter. They're going to have a slightly different letter. This is actually the origins of those letters and the different amino acids with 3-letter and 1-letter codes. So, for example, cysteine right here, the 3-letter code is cis, CYS, and the 1-letter code would be C. Okay? All of these ones, the 1-letter code is the same as the first letter of the amino acid. So, those all make nice sense. This is where things start getting a little confusing. So right here we have arginine, but we've already used A. Can you see that? Alanine was A, wasn't it? So we're in trouble. And so what happened is if you say arginine, right, you actually sounds like you're saying R for the first letter, doesn't it? And so it is R. Right. Over here we have phenyl. Phenyl sounds like an F. And so they put F. We're going to run out of the really good ones in a pretty short period of time, actually. Tyrosine. Tire sounds like Y is the sort of pronounced sound in that. Tryptophan. This is where it starts really starting to stretch, by the way. Tryptophan. And so W. Aspartate sounds a little bit like date at the end. A D sound. And so they put D for that. Asparagine has a very pronounced N at the end. And so they put N for that. Glutamate has a very pronounced E that comes in the name. This also has an E that comes after D. So your D here, aspartate, glutamate. And so D, E comes after D. Glutamine. This is where it really is a stretch. Cutamine. Glutamine. I know. I don't agree with it either. I would not call that a vowel rhyme. Yeah. No. But apparently some biochemist somewhere thinks that those sound similar. I don't know what to tell you there. Lysine. This one's literally, they just took the next letter. So they have an L. They couldn't figure out anything else because they have Y. They have everything else, right? And so it's just the next letter, K. Okay? It's K. Now, amino acids are going to be weak acids. They're also going to be weak bases. That means that they do react with themselves. So when you have an amino acid and it is in a physiological pH, your predominant species is typically going to be this form right here, where you have a protonated amine, you have the carboxylate. Notice that this has a positive charge on one end and a negative charge on the other, doesn't it? Okay. Now, if you don't have any other charges on this, like we have here for alanine, this is called a zwitterion. Literally, zwitter means two-handed. And so it is an ion that has positive and negative, but you have an overall charge at 0 because they would cancel out. Okay? And so that's a zwitterion. By the way, in case you're wondering, Zwitterhinder means two-handed sword. Zwitter means two-handed in German. Huh, yep, you got it. If you're ever wondering, yes, Germany was the center of chemistry after late 1800s to early 1900s up to about 1944. Hint, hint. Mm-hmm. And it's not what you think. It's not the poison gases and stuff. It's actually things like the rocket program. They actually had the most done for semiconductors. They were inventing computer chips, and they invented drugs that were like meth. That's actually a large part of what the World War II strategy was about. Anyways, and then we stole all their chemists. And so chemistry moved to the United States. We got it. Yep. Does anybody know where the rocket program in the United States came from? Okay. Really, really crazy, interesting history of a guy that actually avoided serving any time for war crimes because the United States needed to beat the Ruskies in our rocket program. So, yeah, he was a full-on Nazi scientist. Pretty big believer in the Nazi program during a period of time there. Though I believe he did actually renounce it later on. So, to be fair to that. But there is some wild history about Nazi scientists in the United States of America. And Russia, by the way. Russia did the same thing. Bringing Nazi scientists they captured. So it is some crazy history if you start looking at what's going on there. And look up Social Security involved in that too. You'd be really surprised. All right, so each molecule has a unique pH value at which only the zwitterion will be present. It will be the vast majority of the species present will be the zwitterion. This is actually called the isoelectric point. Notice the P here is kind of like pH, right? Specifically, it's because it's a pH that will be this point. And so it'll be a pH where it is isoelectric, meaning it doesn't have an overall charge, but it has a zwitterion. Okay, so the pi of alanine, for example, is going to have a negative charge at the carboxylate, positive charge on the amine, and that's going to give it an overall charge of 0. The pi for alanine is 6. And so that would be actually the pi. That's where you would have the vast, vast majority, almost all of it, in this form right here. Okay? If you're a pH below 6, you're going to start having less and less of this in this form. And it's going to transfer over to this form. Once you reach pH 2, which is 2.3 right here, that's the pKa for that, you're actually going to have this form as your predominant form. And so that would be the majority of the sample at pH 2.3. Okay? At pH 6, this one's the majority. If you raise the pH instead and get up to pH 9.7, this is going to be the majority of that species. Okay? And so when you raise the pH above 6, we get this being converted to this one. When we lower the pH below 6, we get this being converted to that one. And at pH 6, you're going to have almost all of it in this form right here. Okay? Everybody getting the trend there? Fantastic. Now, every single amino acid does have an isoelectric point. Okay? Now, the charges of these functional groups changes as your pH changes. So the pH values between the pi and the acidic forms is going to cause you to be protonated forms, and so you just have the positive charge. If you go to pHs that are greater than the pi, then you're going to get the deprotonated forms, and so you'll end up with a negative charge. Okay? Now, you may remember we studied condensation reactions in Chapter 5. I know you thought we would never talk about that stuff again. Oh my goodness, it's like I expect you to maintain some sort of information from this course. It is just above and beyond. Okay. So now that we've discussed how terrible your professor is, my goodness, um, awful person. Uh, that's that condensation reaction. You may remember was originally like an alcohol and a carboxylic acid, right? And then you'd have water being taken out, and that's why it's called a condensation reaction. Okay. These amino acids, that's actually how they're going to be joined as well. Okay. They're just going to be another condensation reaction. The only difference here is this time we have an amine instead of an alcohol on one side. Okay. And so it's going to happen between the carboxylic acid and the amine. Okay. Specifically, it's going to typically happen because your biological pH is physiological pH, right? It's going to happen with a carboxylate ion and the protonated amine. And so you're going to have a water molecule that's removed. You're going to have an amide functional group that's formed. If you remember your amide functional groups, they look like this right here. That's the amide. It's the carbonyl, which is the carbon-oxygen double bond, bonded to an amine, right there. And that gives you an amide. These are oftentimes called amide linkages, but you can also call them peptide linkages, okay? Or a peptide bond. And that's because when you make a peptide or a polypeptide dipeptides, this is the link that forms. You are doing condensation reactions. Okay? When you break them apart, you're doing those hydration reactions, right? Where you added water across it. Okay, so that's the hydrolysis, if you remember. Okay, lysing with water, right? Okay, and so this was the condensation reaction you remember before. This is actually how we did things like we joined together like the glycerol and the fatty acids, and we made those lipids. That was that reaction, okay? And they made esters. This is the condensation reaction that happens between amino acids, and this one's going to make amides, all right? And that right there is forming a dipeptide. So again, this is your amide bond that we're forming. It's going to form, in this case, between alanine and valine here. It can form between any amino acids. When you form your linkage here, this can be called an amide linkage, amide bond, but it can also be called a peptide bond. Okay? Don't ask me why biochemists felt it necessary to make an entirely new language for this. But they did, and I kind of understand, because they like calling these peptides, right? And so peptide bond. Fair enough. All right. So in this case right here, this is going to be a dipeptide because it's two, right? It's two amino acids. And so this was a condensation reaction. Notice the oxygen came off here. The two hydrogens came off of this, the nitrogen. And gave you water right over here. Now, you'll notice when this happens, we have one nitrogen that is unreacted. And so this is going to be referred to as the N-terminus. This is the amine group that stays unreacted. Over here, on the other hand, we are always going to have a carboxyl group. This is the C-terminus because it has the carbon there. Okay? And so this is going to be the N-terminus. This is going to be the C-terminus. And we'll just have a nice chain that's developed. In this case, we just have two, which is a dipeptide. Okay? Notice this is alanine, then valine. The three-letter combination is going to look like that. If we do the one-letter code, we can just write it like this, AV. Okay. So again, some of the elements of that dipeptide. We have the N-terminus. This is going to have your unreacted alpha amino group. We have the C, or carboxyl terminus. And this is going to be the unreacted carboxylate group. Notice, because we have these unreacted groups, we could add another one, couldn't we? We could do another amide linkage back here, couldn't we? Okay. These are typically read from the N-terminus to the C. Whenever you're trying to identify the components, you would go through, you would look for the N-terminus, excuse me. Right. Get really dehydrated in the mornings here. Mm-hmm. Yeah, and I've just had my coffee. And then we go put a square around the amino acid at the C terminus, and we give its name. We locate the peptide bond by drawing an arrow to it. That's your peptide bond right there. And then we would put a box around the amide linkage right here, the peptide bond area. This is the amide group. And then we would give the 3 or 1 letter abbreviations for the dipeptide. That would actually give us the dipeptide we're looking at. Now notice that the dipeptides can form between any 2 amino acids. And if we have a pair of these 2 different amino acids, that would actually give you 2 possible combinations. Because in this case, the order matters, doesn't it? This one's going to have a free nitrogen. This one's going to have a free COO, right? And so the order very much does matter here. Okay. And so these are going to be structural isomers. They're going to be different compounds. They're going to have different properties. They would act differently in your body. These are different compounds. Okay. They are isomers of each other. Now, these orders of the amino acids, this is absolutely critical to the structure and function of the compound. If I took my sleeves and my jacket and removed them and tried to, rearrange it, is it still going to function as a jacket? No. The order very much matters, doesn't it? I need the 2 sleeves on opposite sides. If I put them both on the same side, not going to work out very well, is it? Okay, if I put it where my head goes, also not going to work out very well. Okay? And so the structure very, very much matters when we're looking at these things, right? The order is incredibly important in these polypeptides. So any compound containing amino acids joined by a peptide bond can be called a peptide. Dipeptides are the smallest peptides. You can have tripeptides, tetrapeptides. You can go on just like you can like the polygons. I'm going to be honest. I don't know if I've ever actually seen anybody actually use tetrapeptide. Typically, if somebody's going to do those mid-sized ones, they just call it a oligopeptide. And if somebody's going to do the larger ones, they call it polypeptide. And they typically transition early to polypeptide because they get lazy and don't want to say the names of them, right? Yeah. So they just go polypeptide. Yeah. So as you increase the number of amino acids, the compound is going to be referred to as a polypeptide, of course. Poly meaning many, right? Basically, it's when people give up trying to actually count. Biologically active polypeptides can contain 50 or more amino acids. This is going to be called a protein at this point. Up until 50, they're typically called polypeptides still. Okay. Also, another important part is it has to be biologically active. There are proteins that are not biologically active. Okay. It is possible to have a polypeptide that is more than 50 and it's not biologically active. Pretty rare, but it can happen. Now, again, that would be a protein, and that's what you know as proteins. Now, the substance gluten is actually a mixture of proteins. It's found in various grains. This includes wheat, of course. The 2 main proteins of gluten are glutenin and gliadin. This will contain rich regions in the amino acids proline and glutamine. That's what they're rich in. This is going to have a unique amino acid sequence. So when gluten enters the digestive system, some of the peptide bonds are going to be hydrolyzed by enzymes. These enzymes are called proteases. Notice the ACE part tells you it's an enzyme. The proteo part, the proteases, right? Proteins, see that? So they act on proteins. They're an enzyme. Okay. These are going to break down your gluten partially. It's going to leave behind proline and glutamine-rich peptides in the digestive system. Some of the glutamines in these fragments are actually going to be modified by another enzyme. They're going to be modified to glutamate. The enzyme that does this is glutaminase. Notice again, it acts on glutamine, ACE for the enzyme. Okay. That's going to convert it to glutamate. This is going to change your side chain So in glutamine, the side chain was an amide. It's going to go to a carboxylate group. This is going to have a negative charge. Okay. So once you have that negative charge, you're going to go from being polar neutral, right, to a polar acidic residue right here. Okay. So that addition of that negative charge is actually going to change these peptide sequences. This is going to cause people with an autoimmune disorder, which is called celiac disease, to launch an immune response against these peptides. And so you'll get all of the normal immune responses that you'd think of, like when you get sick, localized in your intestines. Okay? And so these symptoms will include things like the inflammation, like how you get inflammation when you're sick. It's going to be inflammation, but it'll be concentrated around the small intestines. Low intestinal absorption of nutrients, because again, you have inflammation, your autoimmune system is attacking the area, and you have malnutrition from low absorption, because you can't absorb it. No matter how much you eat, you're going to get very, very little absorption. And so people with gluten sensitivity should avoid gluten in their diets, obviously. Does anybody actually have gluten sensitivity? No? Okay. It's actually, there's gluten in a lot of stuff. It is not actually easy to avoid gluten. You can. It just has a high cost. Okay? And they kind of have gluten-free everything now, but you have to special order it, and it's like 10 times the cost always. So it's really kind of a brutal thing to end up needing to do. In case you're wondering, by the way, if you go back in the history of wheat, you know how people are like, GMOs, boo, boo, they're terrible. They're killing us all with GMOs. So interestingly enough, we've always been genetically modifying our crops. This is something that's been happening for most of the countries on the planet, most regions, something like 4,000 to 6,000 years. We've been doing it. And if you go back to the original wheat plants, they're actually very short. And if you go back to them, they have very little gluten, actually. And you go through history, and they get bigger and bigger and bigger because Mr. Lord with his, big pointy stick or whatever was saying, get me more crops or I'm going to shove this in you. And so poor farmer person was like, yeah, bigger crop. And so we accidentally selected for higher gluten content because we were trying to get bigger and bigger wheats. So you could theoretically genetically engineer wheat to remove large parts of the gluten content. Yeah. So if you're wondering, yes, we've always been doing GMOs. It's just at the time, GMOs were not done by scientists. They were done by some poor farmer whose payoff was, don't get killed. Don't starve to death during winter. Okay, those were the two incentives. Yep. Interestingly enough, there's all sorts of genetic changes that have happened to humanity due to that farming and a lack of certain nutrients during that process. It's actually kind of a fascinating interplay between humans and development. So primary structures. Primary structures are going to be proteins order. So this is the order of the amino acids we had been discussing before. And this is going to be how those amino acids are joined together. This is going to give you your protein backbone. We typically read these from the N terminus to the C terminus. And so that would be the order that you'd put this primary structure in. Now, notice that an actual protein is not going to be ordered up like that. It's just typically going to be like rotating and moving around space, right? Now, this right here is how we write the side chains whenever we write it as the primary structure. We just put everything dangling off the top here so we can easily identify which amino acids we have. For example, that one's proline. Ooh, so beautiful. Gotta love it. All right, so again, these are the substituents. These right here are the protein backbone. All of these are the side chains over here, the blue ones. Okay, now the secondary structure, this is going to involve the regions that can form things like coils and other repeating patterns of structure within the three-dimensional shape of a protein, often times referred to as motifs, the different motifs of the protein. Anybody know what a motif is? You saw it in like architecture and art, and it's the different patterns you would see. Yeah. It's the designs or patterns you would see that show up in those elements. Those are the motifs, yeah. And so this is going to be very similar. It's different patterns or designs you see in different proteins. Okay? And so the two most common ones that you would see the most often and would be kind of the most ones you would actually kind of know and maybe have heard of before are the alpha helix. This is alpha, by the way, helix. And the beta pleated sheet, which is beta, then pleated, then sheet. Okay. Notice, by the way, there are more than just two. There are a number of these different structures for secondary structures. Okay. But we're just going to cover the two here. Now, the secondary structures in a protein, they're going to be stabilized by all of those molecular interactions we had talked about before. These are going to include things like hydrogen bonding, and hydrogen bonding being the most common one because it's a very strong force and it's perfect for these sort of distances we're going to be looking at. Okay. Now, if you remember your hydrogen bonding, this is not a covalent bond. It's a strong attractive force, and it's going to happen anytime you have hydrogen bonded to an oxygen, nitrogen, or fluorine. The fluorine is less of a concern here. It's oxygen and nitrogen's bonded to a hydrogen that we typically look at. Okay. You'll have noticed that we actually have several sources of these. We have the carboxylate, right? And then we have the amine. And so those are going to be your sources of hydrogen bonding, and most of these. Right here is an alpha helix. An alpha helix is going to be these amino acids are going to twist around in a helical structure. Okay. And what's going to happen is they're going to form hydrogen bonds with the next oxygen above them. See that? You have the amine forming a hydrogen bond with the oxygen above it on each one of these twisted steps. Okay, so that's the amino acid below forming a hydrogen bond with the amino acid above. Okay, this is going to give you a very strong interaction which causes this twist. Okay, now can everybody see how this looks like that? Okay, this is the more common way to see it, which is the cartoon way, because it just makes it easier to look at. When you look at these two things, you may go, hey, that looks hollow. It is not actually hollow. Okay? So the space in between is actually filled with electrons from those atoms. So if you looked at this with a space-filling model, you would see that there actually is no hollow space. You can't go through this. So instead of thinking of this like a tube, it's not a tube, okay? It's like a rebar or like a rope, okay? It's not hollow inside. All right, now, at least no more hollow than anything else is. Now, secondary structure, this is the beta pleated sheets. There's actually several different arrangements of beta pleated sheets, but this is a pretty common one. And so what's going to happen is you're going to have the hydrogen bonds formed across here. So you'll have the polypeptide. It will loop back in on itself, and the hydrogen from a nitrogen will bond to the oxygen here, and vice versa. See that? Back and forth. Okay. And those attractions are going to reinforce that term. And so you get a loop, hydrogen bonding, loop, hydrogen bonding, loop, hydrogen bonding. Okay? This does not have to actually be alternating like this, where it goes back and forth, but this is a very, very common way to make a beta pleated sheet. Now, the reason it's called a pleated sheet here is because if you remember your tetrahedral structure, right? Your tetrahedral structure is actually going to make this zigzag. Okay? And this zigzag is even further reinforced by, see those right there, the purple parts? Those are your R groups. And so what you're going to see here is your R groups are actually going to jut out on the surface of this beta pleated sheet, alternating back and forth. And it's going to make this very exaggerated zigzag pattern. Okay? Kind of cool, huh? Ooh, ah. All right. So again, the chains are going to hang off above and below the beta pleated sheets. Now, you may have already kind of noticed there's some patterns that you may make connections to. Like these beta pleated sheets. What if you had all hydrophobic stuff hanging off of it? Would make like a sticky little mat to stick different hydrophobic stuff to, wouldn't it? Yeah. And so that's actually a very common use for these. They can be these hydrophobic surfaces that things stick to. Okay. These alpha helices oftentimes are structural components. They'll be braided together to make these structural compounds. Okay. Okay. And so let's talk a bit about the secondary structures and its effects on Alzheimer's disease. So Alzheimer's disease, very common form of dementia. It causes memory loss and the ability to think clearly, becoming severe enough to interfere with daily tasks. The disease is progressive, most often occurring in adults over the age of 65. People with the disease have distinctly different brain tissues than people without the disease. If you look at a brain scan, it actually becomes quite obvious. You're looking at Alzheimer's versus non-Alzheimer's. The brain tissue of people with Alzheimer's disease contains plaques between the brain's nerve cells. The buildup of plaques between the neurons and neurofibular tangles inside the neurons are thought to interfere with nerve impulse transmission and contribute to neuronal cell death. I'm going to state here that Alzheimer's is a very heavily researched subject right now. The science is moving really fast over the last couple of years. Not really. Like this part's all mostly correct. Notice how I've been saying contributes to rather than causes. Yes. There's actually multiple facets to Alzheimer's, and so I very specifically avoid saying causes because the cause for Alzheimer has changed 6 times in 2 years.
Yeah. Well, we're learning more and more and more. We need to kind of expand our very previous narrow.
But this is a great example of secondary structure having a play in a disease. No matter what, it's still a great example of it. Because I just have to avoid causes, right? And who knows, maybe in another year or two, we'll be back to causes again.
Well, even when they're saying that it's most often in adults over 65, like they're already showing that we're starting to see differences in the brain in 40-year-olds. And it's just like every other disease, if we can get a hold of it earlier.
Well, the effects of Alzheimer's are most often shown. So the disease is progressive, most often occurring in adults over 65. This is the effects of it that you start seeing. The early indicators for disease, of course, show up much earlier. But you have relatively minor effects from Alzheimer's before 65.
Yeah, let's handle it then, though, before it's...
Oh, of course. Yeah. Of course. Honestly, there's probably a pretty good genetic component to it. It's highly correlated with genetics. And so there's probably going to be like a pre-screening like you do for certain types of cancer. They genetically screen and then you start treating and checking. But anyways, secondary structure. Now, these protein plaques is going to be made up of protein fragments that are 42 amino acids long. They're called beta amyloid protein. A little interesting factoid, they may have actually just discovered a working cure, but they need to do the human testing to make sure it doesn't cause more problems. They've discovered a few things that could have cured Alzheimer's a few times now, but they cause long-term issues. So the protein sequences, these have been identified in both healthy and diseased individuals, of course. A person with Alzheimer's, the structure of the beta amyloid is going to be alpha helical. Person in Alzheimer's is going to be the beta amyloid proteins will form beta strands. So without Alzheimer's, you would see these as alpha helical. Remember the alpha helix that we talked about, right? In a person with Alzheimer's, this is going to form beta strands. So the change is actually going to make the beta amyloid proteins fold into sticky beta-pleated sheets. These will aggregate. This is what actually is going to form your plaques at the neurons. Now, again, this one is currently there isn't a cure for Alzheimer's. There's actually several treatment methods that can delay Alzheimer's and help regain some degree of function. Though for people who have suffered from Alzheimer's for a long period of time, there's probably not much that can be done, because it actually is a very similar disease to like a diabetes, and will actually prevent you from producing energy and eventually cause death of the surrounding area. So it's actually kind of a crazy thing. It's linked heavily to metabolic diseases, in fact. So it's Alzheimer's is a multi-factor disease. But maybe they can figure out one common identifying. So these are some medications. Again, this is one of the reasons why this is a rapidly developing field. Do these medications still work? Yeah, of course. But are they probably the best treatment for it? I would doubt it. Okay? I would always check with your doctor. I would not get attached to some medication I have to put in my lecture notes. Starting a semester, there's probably been, I know of at least 10 major Alzheimer's publications this last semester alone. There's probably been hundreds. Okay? This is a very fast-moving field, so. Anyways, this is one of the treatments you can do is to keep the brain transmitters running high. There are other treatments too involving disassembling of the plaques as well. And there's so many treatments now, I'm not going to try to go into them all. But the most important part of this, for my purposes, is secondary structure. So this is actually directly a secondary structure, right? And so that wonderful example I gave you of secondary structures are alpha helix right here, right? Okay, so normally alpha helix, and we don't have any major problems. We start getting it going into the beta pleated sheets, and we form the plaques. Because notice how these have much better overlap, don't they? Okay? Versus these, which would have very little overlap, right? And so the greater overlap is going to cause plaque formation because they're going to start clumping together. Okay? That's directly the best example I could have possibly given you of why this stuff is important and what sort of effects it can have, right? Look at that. Okay, you gotta love the beauty of that example. All right, so tertiary structures. We've actually hinted at it before. Notice that we are actually changing the tertiary structure in this case right here. And so what tertiary structures are going to be is we're going to have these different parts of the polypeptide chain interact with each other and the environment, and it's going to create a three-dimensional structure, which is the tertiary structure. Okay? The nonpolar side chains, these are going to be repelled by aqueous environments. They're going to turn towards the interior of the protein and interact with other nonpolar environments. Okay? The polar side chains, on the other hand, will be attracted towards the aqueous surroundings and will appear towards the surface on most proteins. And so those will typically interact more with the water, right? Because water is also polar. Now you know why we had to do all that stuff with polar, nonpolar, hydrophobic. So I could talk to you today. Now, the tertiary structure, it's going to be stabilized by all of those attractive forces we talked about before with the side chains. All of them. Every single force we've ever talked about is going to show up now, okay? So, let's look at it. Right here, we have a tertiary structure of a polypeptide. And you'll notice here we have one continuous chain, right? Okay. And so the tertiary structure is the three-dimensional structure of one continuous chain. Now, this right here is an ionic salt bridge. See that? We have an ion-ion attraction, don't we? And so that's called a salt bridge. This right here is a hydrogen bond. There's another hydrogen bond, more hydrogen bonds. This right here is a hydrophobic interaction, which creates that sort of turn in it. And over here we have hydrogen bonding to water, which will allow this to solvate. And over here we actually have a covalent bond. Now, these ones are kind of unique. They're what's called a disulfide bond. Okay? And they happen where we have those sulfides at the end. Remember the SHs we were talking about with cysteine? Yeah, they happen there. They form the disulfide bonds. And so it'll covalently keep it together. Okay? Now, these tertiary structure interactions are going to have all of these different interactions we've talked about before. So there's the salt bridges. These are going to be the ionic interactions. And again, when we have these opposite charges attract within that protein, it's going to create our salt bridge. They're a little bridge with ionic attraction, right? They'll keep the thing together. We have the disulfide bonds. This is going to be two thiols reacting together. And it's going to cause a disulfide bond. Now, one of the things I would like to point out is covalent bonds don't normally break and reform a lot. But finding conditions you can break disulfide bonds is not that unusual. Okay? So, yeah, they don't under normal circumstances break a lot, but you can actually cause them to break. Okay? And many of you have probably actually caused your disulfide bonds to break. And we'll talk about it in just a second. But this right here is your thiols. This right here is your disulfide bond. Notice that this is an oxidation over here. This is a reduction going the other way. Okay? And so if you put these under oxidizing and reducing conditions, you can cause these to break or reform. Okay? This is an equilibrium you can actually play out. So why am I talking about breaking and reforming disulfide bonds like you all do it all the time? Okay, anybody ever had a perm or a hair straightening done? Okay?
Chemically or just with a hot iron?
Either.
I mean, I use a hot iron.
Yeah. It doesn't matter either. Chemically, it will just be the oxidizing, reducing conditions.
Okay.
If you use a flat iron, you're using heat to do it.
Mm-hmm.
But what you're doing is you're breaking and reforming disulfide bonds. That's what gives you the curl in your hair. Okay? So if you have curly hair, disulfide linkages, these disulfide bonds, you got lots of them. Okay? If you have more straight hair, you have less of these. Okay? And when you're straightening your hair, you're obviously reducing the disulfide linkages. If you curl your hair, on the other hand, or kink your hair, you're increasing those disulfide linkages in that specific area. Okay? And so, ta-da! We have our disulfide linkages that you've all had and been manipulating and doing stuff with, right? All of this time. You didn't even know. Okay, so this right here is a tertiary structure, very commonly represented as little cartoon structures because it makes it easier to see. You can see this one has a lot of alpha helix secondary structure, right? And see how it's like this little globular shape? Looks like a little kind of globe or like a glob of something. Okay, so that's called a globular protein. That's the description of that tertiary structure. And so this is going to have your N-terminus right here, C-terminus right here. Exterior part right here is sort of minimized. There's going to be a lot of like hydrophilic groups on the outside of this. And this is going to allow it to face water and maintain solubility for a period of time. It won't actually dissolve in water, but it will be able to stay in water for a fair amount of time. Okay? As long as it's agitated and moved around, it'll stay in water. Okay, so you can also have fibrous proteins. And so right here we actually see an alpha-carotene coil. So this is an alpha-carotene coiled coil. Anybody know what this would be? Hair. Yeah, this is your hair. Okay. This right here is a collagen triple helix. And this is typically found in your skin. Would probably be the one you'd think of the most. These are actually found in a variety of different things, like horns and wool and hair, and this one nails and yeah. So again, these are both alpha helixes. Actually, you can see they're just braided alpha helices, and that gives you the structural component. Okay. And they're fibrous, and that would be your tertiary structure. That's fibrous. Now, having said that, did any of you realize that you've actually seen individual proteins before in your life?
Mhm.
Yeah, that was your hairs, wasn't it? Those are individual proteins. All right, so your quaternary structure. This is going to describe interactions of two or more polypeptide chains to form a single biologically active protein. So remember how when we were on tertiary structure, I stressed that's a single polypeptide, one continuous chain, right? So if you have multiple of them joined together, this is going to give you your quaternary structure. And notice that not all proteins have to have a quaternary structure. Okay, some do not have a quaternary structure. Right? And to have a quaternary structure, you must have more than one polypeptide chain joining. Okay. Now, keep in mind that these individual subunits, which would be the individual parts of that quaternary structure, these are going to be held together by those same interactions you would see for the tertiary structure. So we're not actually adding anything new. It's just, in this case, we're going to have multi-polypeptides joined together. Okay? Rather than just having it just be one folded up. So, okay. Let's go ahead and look at this in terms of figures. So this would be your primary structure. It's the order of your amino acids. They're going to fold up into a shape. There'll be repeated patterns, which will be a secondary structure. Those repeated patterns join together into a tertiary structure, which is a larger structure. And then they join together into a quaternary structure. Okay? And then we get the full structure of our hemoglobin right there. All right? You can see hemoglobin. See those little pieces right there? Those are your hemes for hemoglobin. They're where the oxygens bind. Ooh, ah. It's a globular structure again, by the way. Okay. Well, that has a lot of area, doesn't it? Okay. Notice it looks a bit like my cords in my bag, doesn't it? Where I've kind of secured the cord around something and then pushed it over to the corner. Okay, what happens if I unravel all my cords in my bag? They all end up tangled together, don't they? They end up with this single mass of stuff I can't really separate easily, right? And so guess what happens when you cook food? You have your nicely ordered, tertiary and quaternary structures, like your egg right here, right? And they're floating around in solution. And so it's liquidy. And then you start cooking it. And what happens? Well, you denature your proteins. You cause your proteins to unravel. Because you start breaking all of those attractive forces, right? And remember, we can boil water and we break the polar bonds, right? But we don't break the covalent bond, do we? And so in these proteins, when we provide just enough energy, we're going to start breaking all of these polar hydrogen bonds, salt bridges, those sorts of things. But we won't break the covalent bonds. And the covalent bonds are what keep those amino acids ordered. Now, what happens if we cook something at too high of a temperature, by the way? We burn it. What is burning? It is yucky. Burning is where we break the covalent bonds. So we destroy the amino acids and we destroy the primary structure. Okay? If you're just destroying the secondary, tertiary, quaternary structure, that's denaturing. Okay? And that would be what we consider cooking usually. Okay?
What about when you use salt to cook something like a ceviche?
You got the next part.
Okay.
You were exactly right. Yeah, so when you use salt, actually, what's going to happen is you're going to disrupt that hydrogen bonding and your salt bridges.
Mm-hmm.
And again, you're going to denature your protein. And so that's one way to denature proteins. You can also use acids and bases to denature proteins. That would be things like, lactic acid from bacteria is actually how you do yogurt and cheese and stuff.
Yep.
But you can also use vinegar to do it, in which case you get mayonnaise, right?
Yeah.
Yeah. Heat, you only need temperatures above 50 degrees, by the way. That's why you can slow cook things.
Yep.
Yeah. 50 degrees is going to allow you, 50 degrees, by the way, is not very hot. 50 degrees is like
You don't mean Fahrenheit, you mean Celsius.
Celsius.
Yeah.
Yeah,
Okay.
yeah, not Fahrenheit. No.
Yeah. I was like,
no, no,
now we're almost at refrigerator in the 40s.
no.
Okay.
Now the other way you can do it, you can actually use reducing agents to partially denature things. And so this would actually be your disulfide bonds that you would actually reduce. And this is actually how you can straighten, and then you can cause them to reoxidate to forms you want them to be in. And give yourself like permanent waves or, super curly or whatever you want. Okay. This is why you actually need reducing agents if you want to go like the hot, like longer lasting ones that are more extreme. Yeah. You typically need a little heat too, though. But this is what they're doing when they do that paint stuff on it. You all know what I'm talking about, right? I've never had it done to me. You can tell I haven't had it done to me. I don't even know what it's called. Now, heavy metals can also do this and disrupt your disulfide bonds and your salt bridges. And so, for example, mercury and lead poisoning do this in your body. Typically, you don't use this for anything because we don't want to increase the amount of heavy metals in us. And so you would not cook like this because it'd be a problem. But what you're talking about with the metal, just using salts, it does the exact same thing. It just doesn't do it as effectively.
Right.
Yeah. And so you can do large concentrations of salts to do something very similar. Mechanical agitation also works. You'll actually break hydrogen bonds and London dispersion forces. And so, for example, you can do like whipped cream, right? Take cream and whip it and get whipped cream. You can also take cream and churn it, which gives you butter. Again, it's a mechanical agitation. And meringue would be egg whites where you whip them. So those are all mechanical agitations giving you denaturation. So, by the way, these same things work with like detergents actually do this as well. And so they actually interrupt the hydrophobic interactions. Obviously, you would not want to cook with a detergent. You could, but it would not be recommended. In fact, if you ever want to do something kind of fun, take a small amount of milk, put detergent in it, and you'll see it go whoosh. Yeah. It'll do it really fast. And what's happening actually is you're causing denaturation of your membrane proteins. That's why detergents can kill bacteria and viruses so well. Is because they actually denature their proteins.
But vinegar. We use a lot of cleaning vinegar.
Yep. Vinegar still works. That's the acids right here.
Yeah.
Yep. And yep, vinegar does work. Especially if you put like a salt with it. Because you can actually tackle most of the different things that you would find in these interactions.
So could I dissolve salt in my cleaning vinegars and make them more effective?
You can. Yeah. That's actually like, you've never heard of salt vinegar?
Yeah, scrub with it, but I've never used it as like a bacterial cleaning just to leave it there.
Oh, yeah. Yeah. Now, the one reason you may not want to do it, though, is because then some of the salt could be left behind if it's not soluble enough. Yeah. And so...
potentially be caustic from the countertop, too. Like, salt will definitely erode concrete.
It depends on the salt, but yes, it will cause certain things to become more soluble. Yeah. That actually is something I just taught in different higher division class about why that happens. Okay, so this is actually the hair relaxing and perming that we were talking about. And so right here you have normal curly hair, and so you break your disulfide linkages here in your normal curly hair. This is going to give you a straighter hair, and then you can actually reform them by using an oxidizing agent into kind of whatever we want to do. You could go curlier, you could go wave, you could go whatever you want. I don't know. What's other popular with you youngins? Or fashionable people? Obviously not. I'm wearing a Costco coat. They're nice. I don't know if they're fashionable or not, but they're nice. All right. Now, one of the treatments you can actually use if you ever get heavy metal poisoning, by the way, and you could actually self-administer if you had to, but I would highly recommend going to a hospital if you get heavy metal poisoning, is like lead and mercury poisoning. You can actually ingest a bunch of egg whites. Those proteins will end up picking up lead and mercury and reduce the amount of lead and mercury in your digestive tract and then your blood, obviously.
Raw or cooked?
Raw would work better probably,
Yeah.
but I guess you could probably get away with doing cooked as well. But you would have already disrupted a lot of those salt bridges, so you would have less available. Yeah. Now, in the medical, as far as I remember, they still use EDTA to do this, which is a much more effective. It's called chelating, by the way, where you bind metals to things that will hold them really well. And so it's called a chelating agent when you do this. And EDTA is a much more effective one usually. But there's a variety of chelating agents they use in the medical industry. So I'd have to look and see if they've changed. All right. By the way, you would then take something to cause vomiting, to induce vomiting, because you would want to remove the heavy metal from the environment. You would not want to leave it in there to possibly redissolve when you digest the proteins. Okay. All right. So this right here is some different protein functions. And so they can act as messengers, receptors, transport, storage, contraction, obviously in your muscles, right? Protection, structure, catalysis. These are probably what you mostly think of proteins as, which would be enzymes, where they're acting as catalysts. But you can act as many, many different things. For example, insulin is a messenger. There's the insulin receptor, which is a receptor. Both of those are proteins. Right here, we've already seen some different proteins acting as various different parts of this. So we have a hormone, this is a chemical. Sometimes it's a peptide or a protein. Sometimes it is not. It'll be created in one part of the body, and it'll affect another part of the body as a message. Receptors are going to be proteins. These will face the outer surface of the cell. They'll bind to a hormone or other messenger, and they'll trigger a signal inside the cell. We also have the transporters we talked about before, which are going to be integral membrane proteins. These will span the phospholipid bilayer. Very commonly, by the way, these are alpha helices spanning the phospholipid bilayer, but not one. It's like a braided. You ever seen one of those braided little tunnel things? Very similar to that. Okay. All right, so this right here is going to be an insulin receptor right here. We'll bind to the surface, and then we'll release some sort of signal inside the cell. This one right here would actually be a glucose transporter. Remember, these can be passive or active. Active would use energy to do the transportation. Okay. This is actually hemoglobin. So this is your body's oxygen transporter. We had talked about numerous times before at this point, right? We come back to hemoglobin all the time. Again, this hemoglobin is going to have 4 subunits. You can see them in blue and red here. See those? So it has 2 sets of 2. Okay? And this is the quaternary structure right here. So each of these heme groups right here is a prosthetic group. That's that little green part with the little metal ion bound to it. That's an orange little metal ion. That's an iron. And those are actually what bind the oxygen in the hemoglobin. Okay? This form right here is actually the oxygen on-bound form, the deoxy state, which doesn't have oxygen bound. This down here would be the oxygen-bound form, the oxygenated state. You can see it actually changes structure. Can everybody see that? See a little hole in it? It actually tightens down. So the interesting thing about hemoglobin is it actually undergoes a conformational change when oxygen binds to it. So when the first oxygen binds this hemoglobin, it'll start changing towards this form here. This form actually binds oxygen better. When oxygen starts coming off of this, it's going to change to this form here, and it'll actually bind oxygen worse. And so what it'll do is that your lungs, they'll start taking on oxygen, it'll close in, and it'll take on a bunch of oxygen. And all 4. And then it will go off in your cells, it starts losing an oxygen, it opens up, and it loses all of it. And so it dumps all of its oxygen into your limbs. Okay? Into your extremities. There's kind of a cool thing. that you actually have a little part in your blood that can actually fit in this space right here and act like a little wedge. And so it can literally go like bang, wedge in there and cause it to release oxygen. Okay? So there's some cool stuff you have going on in your body there. Kind of interesting. Anybody have any questions? That's a conformational change. In case you're wondering, if you're wondering what an oxygen carrier with only one subunit looks like, that would be myoglobin, which is what you actually find in like meats. That's the thing you probably thought was blood. It's not, it's myoglobin. Okay. Next time around, nope, we're still talking actually. We have 2 minutes. Why you guys always prep? It's gonna start going. You know I'm not gonna end. Now, antibodies. So this is going to be your body's defense system. So when foreign substances, these are going to be called antigens, like bacteria into your body, your immune system is going to produce substances that we call antibodies. These are going to be proteins. So an antibody is going to consist of 4 polypeptide subunits. We have the 2 heavy chains, which are the 2 large portions right here. And then we have the 2 light chains, which are these parts right here. That are the lighter ones. Okay? And so our secondary structure is actually going to contain beta-pleated sheets. These are these flat ribbon parts right here that will clump together and be attracted to each other. And so these are going to be stacked very tightly to each other. Your quaternary structure is actually going to be held together through disulfide linkages, disulfide bonds. Right here, we can see the disulfide linkages and bonds. And so this is actually going to give us a Y shape like this. The antibodies are going to bind antigens at this point right here. These are actually the binding sites that will actually identify the antigen, which are your invaders. So once they bind, they actually can then split, and that's going to give a signal that, hey, this thing needs to be attacked. Come, attack this. Okay? Now, it's kind of interesting that these points right here, which are these binding sites, can actually be geared towards binding almost anything. You can bind all sorts of things with these. You can actually design them to bind things like drugs. And so you can actually make like these antibodies that will bind drugs and cause the drug to flush from the system, or will identify the drug. Yeah. You can do all sorts of kind of cool stuff. We'll start here next time. You all have a wonderful day. What do we have coming up? The exam.